Hey y'all, as you can see, I got my lab coat today, and I got my gloves, and these are important because we're going to be going over aseptic technique, and it's important to keep everything clean in lab so you don't get any contaminations. So, the first things first is that when you walk into lab, you want to make sure to wipe down your station with Vespine. I know a lot of y'all haven't been doing that. But you want to clean your station. Vespine's like on the top. You just spray it on the lab bench. Then you take napkins and then you wipe them down, and that ensures that everything is clean. Next thing is you want to wash your hands as well. And then if you're a girl, you want to make sure to put your hair back so nothing like falls into your stuff. And then roll up your sleeves. Just make sure nothing touches anything that you don't want it to touch. And okay, so that's some stuff that some of y'all haven't been doing, but so try doing that. There's more of those type of things on page 19. If you read the background information, you'll discover some more of them, and that's important. But for right now, we're going to be going over how to make a slide for viewing under the microscope. So right now, I got my broth culture. It's lemon juice, but it looks like broth culture. I got an inoculating loop, a Bunsen burner, and a glass slide. And so that's all you need. So the first thing you do is, when you're doing aseptic technique, is to make sure you label your slide. In the practical, you're going to be doing a gram stain, and so when you do a gram stain, it's going to be a little bit different with all your staining. So, but you're going to write like on your slide, like your name, the date, section, bench, bacteria you're putting on there. Maybe put gram stain or something. Just make sure that you label it to where you can easily identify what is on here. All right, but when you go to draw your circle, some of y'all have been drawing really small circles. What I do is I draw the biggest circle as possible, so I touch the edges, and what that does is that when you go to smear your stuff on the slide, it spreads everything out, and it also will dry quicker because you have a thinner layer of liquid, and that when you spread it all out, it'll spread out the bacteria, because some of y'all have been getting clumps of bacteria, especially if you're taking from a plate. You want to make sure to spread them out so you can easily identify the cellular morphology, because that's typically the purpose. If you're doing a simple stain, if you're doing a gram stain, you can differentiate some other stuff. But when you're staining, it allows you to see the cell morphology, and when they're clumped up like that, you usually can't see it. So spread it out, make a bigger circle, spread it out, and what that'll do is let you have a more successful viewing underneath the microscope. So the first step is how to hold this stuff. He didn't really go over it very well, but what I do is that you have your inoculating loop in your right hand, or your dominant hand, I'm going to say, and then your culture in your non-dominant hand. So what I do is I take the pinky and then I grab the top again, and that allows you to still work with everything, because you can kind of work with the pinky, but all right. But when you're holding the tube, you want to make sure to hold it at an angle, because what that does is that it kills the bacteria, or it doesn't allow the bacteria to fall in the tube as easily as if you hold it like this. This bacteria just in the air can fall easily into the tube and then grow and then contaminate the culture. But if you hold it like this, it's harder, at least. And then you pass it over the flame if it's glass. If it's plastic, do not pass over the flame. I know he said in class that you do it anyway, but you don't. It says in the top right corner on page 20 that if the tube is plastic, it should not be passed through the flame. So do not do that. But if it's glass, go ahead, pass through the flame. That kills some of the stuff that's lingering in the upper region of the tube. And so you pass it through, still hold it at an angle, and then burn your inoculating loop. So some of y'all have been burning it like this, which I guess works. If it glows, you're sterilizing it with the heated culture thing, and so, but if you hold it like this, you the flame can like kind of travel up the loop and get all of that kind of in one pure swoop of flaming. And so try to hold it like this. And then what I do is I shake it and I count to 5. So I like 1, 2, 3, 4, 5. And then usually it's cooled by then. If you don't allow it to cool when you put it in your broth, it'll like sizzle, and you don't want that because that'll kill some stuff. Which there's a lot of bacteria in here, so it's really not that big of a deal. But you want to try to make sure to cool it off to make sure that it doesn't that you can still get some stuff. So you put it in there, and you kind of shake it around, and you grab a loop full of culture. Then you take your glass thing and you flame it. If it's plastic, you just kind of don't flame it. And then you put your lid back on with your pinky, and then you put that back in wherever it goes. All right, so now you have a loop full of culture and your labeled glass slide. So you take your loop full of culture and you spread it around as much as possible. Try to stay within the circle. If you find that there's still too much on that, you can if you're careful, kind of go over the circle and spread it out. It'll dry quicker. But again, if you make a big enough circle, you're good. So you let this dry until you can't really see. Like if you like hold it up to the light, there's no like glare on it. And you let it dry, and then you'll end up heat fixing it. So you pass it over the Bunsen burner twice, maybe 3 times. Try not to do it over that because you don't want to overheat it because you can kill some of the stuff on here. So like 1, 2, 3, and then you should be good. Don't hold it directly over it because that is a recipe for disaster. And then pass it through. Also, you don't want to be touching your slide. You want to make sure to use your little clothespins that they give you in your drawer when you're doing that, because the glass does get hot and you don't want to like drop it. You don't want to like burn your hand and then drop it, and then just use your clothespins. And then so that'll heat fix it, and then you're ready to do staining techniques. In this lab we did a simple stain, but on the practical you'll be doing a gram stain. So we'll be going over gram stain in a little bit, and we'll so that's aseptic technique. Try to correspond to that as much as possible.